And I've got top of the hour here, so I'm going to get started. I want to first thank everyone for joining and wish you all a good morning, good afternoon, and a good evening. Thank you all of you for joining today. My name is Rahul Madhavan, and I'm here with Point Cross. And we're going to be diving right into our webinar focused on an efficient and an accurate and a future-proofed mechanism to manage and standardize disparate biomarker data. And this is actually the first in a series of webinars where we're going to be hosting, and this session focuses primarily on the cell phenotype domain. Before we start, a key reason for hosting this event, it's almost like a lessons learned session from the wide range of sponsors and the CROs that we've had the opportunity and privilege to work with, who may have statistical programmers or standards managers or other analysis dependencies that rely on genomic or protein or cell and tissue biomarkers. And what we see is a trend where complex, novel assays are sometimes not standardized unless they explicitly inform a CSR-defined endpoint. And ultimately, this affects data reuse, integrated and holistic patient data sets for analysis, and indeed, the robustness of digital regulatory submissions. Point Cross has been a long-standing member of the data standardization and data management community, and we have over the last few years really doubled down in our focus on standardizing biomarker data. The data that's often described as external data or disparate data or central lab data or unstructured data or exploratory data or assay or translational or multi-omic data, however you call it. Fundamentally, when you look at our history, you can see that there's been an intentional, gradual approach to handling the complexities of natural data through our multimodal XBiome platform. So a quick housekeeping note, we welcome questions throughout today's session. So please do use that Q&A button on the bottom of your toolbar to ask a question if it comes to mind, or if you're watching this recording, please drop an email to us at hello@pointcross.com, and we'll be sure to respond to you with our answer. So without further delay, I'm really pleased to be able to hand this over to Arpita, our senior subject matter expert, who's going to be giving us a walkthrough on our approach to helping sponsors and CRO customers streamline the management of their natural and external data flow. Thanks so much.
Thanks, Rahul. Hello, everyone. Welcome to our webinar today. So I am Arpita, leading subject matter expert, leading data standardization of clinical and translation data at Point Cross. So today, in today's webinar, I will be walking you through the approach of flow cytometry data standardization through our XBiome tools. So part of the webinar agenda, we would be talking about giving a little introduction about the flow cytometry assay, the usage of it, and when it comes to submitting the data to regulatory submissions, what are the guidelines to be followed, and what are the current challenges being faced, and how XBiome platform can overcome the standardization challenges. So let's begin with a little quick introduction of flow cytometry. So flow cytometry is an assay method where it uses the laser to examine the physical and chemical properties of each cell flowing through a liquid stream. The flow cytometry is a rapidly used assay because it gives very high insights on each cells through identification of the markers that the cells are uniquely characterized to. It has been majorly used by the pharmaceutical industries, specifically during the drug development, the vaccine development, and majorly in cell-based therapies to monitor the immune responses, the drug effectiveness, and examine the impacted cell populations. So as this assay is being widely used, so it has to be submitted to the regulatory standards. So when it comes to regulatory submissions, for FDA, EMA, or PMDA, the flow cytometry data has to be transformed to CDISC compliant formats where the markers, the measurements has to be aligned and represented accurately as per the CDISC guidelines. During this standardization process, there are very common challenges faced which are interpreting how the lab provided reportables are to be standardized and mapped to the global registries or CDISC terminologies, and identifying the key variables, which includes the marker string, the sublineage information, the cell state markers. So these are the key variables which has to be identified accurately and represented, and understanding the gating strategies employed. The flow cytometry, as known, heavily depends on the gating strategies to specifically isolate the cell populations to measure. So interpreting that part of the standardization requires thorough understanding and interpreting that information, and deriving the metadata. So the metadata calls depending upon the measurement types, whether the cells are being measured for an absolute count or a relative measurements, or intensities are being measured, which is a single cell expressions. So each of the measurement types has different metadata, and that has to be represented accurately, and then following the guidelines for the data representation. So for each of the measurement type, there are certain guidelines that has published by CDISC to represent the data. So all that has to be followed during the standardization process. And finally comes the cross collaboration between the lab scientists and the data providers to accurately identify which information of the assay has to be represented in the data sets. So these are the common challenges faced during the standardization process. So we are as we know, we have discussed about the challenges, and let's see what are the guidelines that has been set up by CDISC. So CDISC has the CP domain, the cell phenotyping domain of SDTM, has certain key variables defined, which would uniquely represent the test or cells being measured. These are the variables like the test CD markers, sublineage markers. So these are the variables which uniquely identify the test or the measurement being assessed. So the test and the test CD variables are the key variables where the cell population or the cell name has to be standardized. And the marker strings and the sublineages are the variables where the surface markers which characterizes the cells has to be represented as per the lineage defined from its parent population to the subpopulations. And there are during these assessments of the cells would be in different status, and there are key markers which identifies the functional state of the markers, and those has to be represented in variables like cell state markers. So having these variables and the representation of these are the guidelines that's provided. So let me walk you through now the XBiome approach of streamlining the standardization process. So the XBiome metadata repository and the smart transformation module allows to simplify and harmonize the complex biomarker data, having access to the global registries and the control terminologies. What this means is the marker string will be standardized irrespective of different assays, different vendors, or different panels. The markers would be consistently standardized. And the representation, what the figure illustrates the representation of the cell populations in MDR. So each cell populations and its respective corresponding cell markers are tagged, and they are hierarchically arranged as per the lineages. And these lineages are constructed in MDR using the global registries like cell ontology and cell markers. So during the process of standardization in XBiome tool, what exactly happens here? So there is a series of validation checks, and the standardization and aligning of these markers happens. So as the marker string provided by the reportable is imported into XBiome, it will be validated, and after the validation process, the marker string would be reviewed and aligned to a global markers. During this stage, each of the markers in the string would be identified and classified to identify whether these markers belong to a parent population or they define subpopulations are their markers which denotes the structural or functional state of a cell. So it processes each of the markers in the strand and categorizes as the parent, child, and so these characterizations or the classifications helps to ensure the consistency during the standardization process. Along with these information of identifying the markers and aligning to global registries, there are essential variables like the gating strategies, where the gate and gate definitions would be sourced from the assay validation report and also be recommended by the tool by identifying the lineage and predicts the possible gating strategies. Let me walk you through an example of how an absolute measurement and a relative measurement would be processed, and then I would show you the tool. So this is an example of a reportable provided by the vendor, where it is denoting the numbers and indicating the positive and negative expression of it. So when this is sent to the tool, it would first align to the markers to the global standards by identifying the markers. So if these are the numbers represented, it aligns to the CD markers of it. So once these alignments are done, it then processes each of the markers to identify the category, and then it would now align to the global markers. So this is the lineages that is recommended by the tool. So there could be a number of markers attached in the string. So depending on the lineage, it recommends a set of global markers, and these global markers would be then transformed, mapped to the CDISC CP variables. So this is the process that happens, and this is how the output would be mapped. The global term that is recommended aligns to the representation of the CDISC variables, whether it is marker string, sub-marker string, or other variables. It aligns as per the CDISC guidelines. This is an example of an absolute measurement. When it comes to relative measurement, that is cell being measured with respect to another population of cells. So in this case, in this kind of measurement, CDISC has a defined guidelines to be followed to represent that measurement and the uniqueness to be maintained. So the processing is the similar way as it aligns to the global markers. And while representing that to a CDISC standards, it takes care of those guidelines provided. When it is a relative measurement, it takes care of what is the population of cell measured with respect to the population it has been measured. So these standard and in the process of standardization, these steps would be carried out and aligns to the CDISC standards. Let me just walk you through the tool. So this is the standardization tool. So I would just show you an example of a set of vendor reportables being processed and how the tool identifies the lineages and predicts. So this is a string of markers coming in, and so these are the possible recommendations, and it gives a scores of what are the high prevalent string that has to be considered. So it splits up depending on the markers, what each belongs to. So this string has a specific state marker attached, so it identifies it and captures that information. And in cases, it recommends the gate and gate definitions. So during the standardization process, there are certain checkpoints to ensure the data standardization process is streamlined. So when the marker string provided by the vendor is passed into the X biome, it validates the reportable and identifies if the string is good to go to the next stage. If there is any issues being identified, it would prompt through a workflow where the subject matter experts can review those marker strings and take the best next step during the standardization process. And once the validation is completed or it is passed, it later on go to the standardization process where the markers would be categorized and aligned. So during this process, there are chances where a set of markers can be recommended to multiple cell populations. So in that scenarios, the workflow would be prompted, a decision-making workflow will be prompted where user can resolve the issue based on the inputs provided by the tool or can call for an expertise review by the scientific lead or the biomarker lead and take necessary actions. So these are the validation checks happens. So when a reportable is being sent to the XBiome, it validates for its accuracy. So this is a string that is sent and it is been validated, telling there are gaps in the reportable send because it is having the markers of two different populations which are mutually exclusive. So these kind of review and flagging would be done where the subject matter experts can review these and then take necessary actions by having the collaboration between the scientific and the lab, the vendors who provided the data. So in the stages where during the standardization, if there are multiple recommendations that's generated, it again prompts for a decision-making workflow where the tool prompts for a particular string of markers. There is multiple recommendations that is generated, and it asks for a expertise review to choose the correct definitions for this particular reportables and then proceed for the standardization. So what these checkpoints makes, so these checkpoints allows you to standardize the data consistently and ensures there is the governance maintained from the start and end of the standardization process. So the outputs that's generated by the tool would be as per the CDISC structure. So validating the compliant of the outputs that is being generated by the tool would be performed by the XBiome EDV tool, which validates the outputs as per the global rules released by CDISC, FDA, and PMDA to ensure the transformation is CDISC compliant. Apart from the global rules that is available to validate the datasets, at Pointcross, we have developed certain proprietary rules to validate the input strings provided by the vendor as well as the outputs generated by the tool to ensure the outputs are not just compliant to as per the CDISC, but also ensures the data representation follows the CDISC guidelines for each of the data types that is standardized. So these are the set of rules which validates the input markers provided by the vendor. And these are the validation rules that were built to validate the CDISC output to make sure the guidelines, data representation guidelines are met in the outputs. So this is a screenshot of how what kind of inputs goes to XBiome with respect to the float data. So the string of markers and the unit of measurement. When this information is sent, this will undergo a standardization process and generates the CP domain as per the CDISC compliance. So this output that is generated by XBiome is ready to use for your regulatory use as it aligns the compliance and the guidelines as defined. So to have a conclusion, XBiome platform simplifies the standardization process of diverse biomarker types, making sure the standardization process is efficient, compliant, and the outputs generated are ready for your regulatory use. Thank you.
Thanks, Arpita. We have about 10 minutes or so. So we have a couple of questions that have been asked so far. I'm going to read one out to you if that's all right. And it asks if you could describe how you work with the sponsor to receive the central lab data exports. Does this mean that you work to define the data transfer specifications, or does it not matter what kind of export file is received?
Thank you. There is no defined format. The Smart Transformation tool of XBiome can support and accept all varied formats. If there is a specific DRT available by a sponsor, that is acceptable as well. So to tell, there is no specific format. Any formats is acceptable by the tool.
Okay. Another question here is, could you describe what some of the conformance rules look like that Point Cross has defined? I'm assuming that that was the extended rules you referred to in the eData Validator platform to ensure compliance output.
Yeah. So these are the rules. So if I have to explain one or two rules of how this is defined. So when it is about input string validation, so we would validate the consistency of the markers represented. So there are scenarios where we have experienced where the markers are being defined, expressing both a negative and a positive expression, which would never be a case except when there is a mid expression reported. Else, it is always a positive expression or a negative expression. When both are being reported in a string for a specific marker, that's a violation, and that calls for a validation checks or a prompt review. And similarly, there are also scenarios where the markers would have no definition of from where these subpopulations are defined from. So without a parent lineage available, it would not be possible to define a CDISC equivalent test names. So those are the kind of input checks that happens. And similarly, when we have the rules for the output checks, so there are certain variables which are like a paired variables. If one exists, the other one has to be there. So such kind of checks, example of gate and gate definition. If a gating strategy is available, we should be able to standardize both gate and the gate definition variables and the representation of the data. So the representation of the marker strings or a test names are specific for a kind of measurements. If it is a relative measurements, both the test and the marker string has to be denoted that they are a relative measurement. And similarly, if it is a fluorescence intensity measurement, the marker string has to be represented with the units that is being measured, whether it's a mean or a median or just a fluorescence intensity, that has to be captured as part of the marker string. So these are certain guidelines, and to validate that data is represented as per the guidelines, these rules are developed.
Great. Thank you. There are two more questions, and I think that that should bring us to the top of the hour, but I do welcome if there are any more questions. We'll be responding to any written questions in the chat box there. We'll be responding and putting that both on our LinkedIn as well as on our website in response in writing. So we do welcome any continued questions that come, or even after this, if you have any, you can drop us a line at hello@pointcross.com. But one question we have is, do we have the ability to handle receptor occupancy assays?
Yes. Yes, in today's webinar, we discuss more about the cell phenotyping. So we support the receptor occupancy part of the CP domain, yes.
Okay. How does the tool standardize those test names that are not defined in the CDISC standard?
As described, the approach of standardizing the vendor-reported markers is to the global registries. So the global registries, as mentioned, are built based on the global databases like cell ontology and human cell markers, where there are cases where a specific cell population or a test name is not defined in the CDISC yet, but standardizing to global registries still has a global term attached, and the tool recommends a test name based on the global term, which can then be reviewed by the subject matter expert and consider it as an extended term.
Got it.
There is always a recommendation to the global terms. So you always have a cell population attached during the recommendation.
Okay, perfect. Thank you. We have a question here on pricing and whether we have a question that's just come in on whether we handle results data only or at the metadata level. Can we manage standardization at the metadata level? I assume that would mean for downstream biometrics handoff. Could you maybe take the question on upstream, downstream, and I'll take the pricing question right after that.
Yeah. The standardization is done at the metadata level as well as the results level. So the standardization here is more about how best you align the markers to the CDISC compliant format. So the standardization supports on both aspects of metadata and the subject level data.
Okay. And the question on pricing is, how is this priced? So I think that's pretty straightforward. The answer is also pretty straightforward. Assays themselves, if there are at the protocol definition stage, or if you have certain assays that you're looking at running or that you know have been run and need to be standardized, the standardization and the ability for those to be rendered into the CDISC compliant format as standardized within the CP or later on the GF or other specific domains that come about within SDTM, those are just a couple of $100. Really, it's on a per assay basis. That's completely acceptable and normal. And a lot of the CRO-based customers come on that kind of a basis. Sponsors tend to think of the biomarker data set as a sub-repository within a broader clinical data set. And the way that we've engaged with them is to create a repository that's, number one, queryable, and number two, the ability to delve into more of a robust or holistic analysis of the patient data set, wherein the exploratory biomarker data is integrated with EDC or ECRF data. And once that's accomplished and that patient data integration occurs, it's not dependent on necessarily an SDTM output, but rather there's an invariant data model that we have, a repository data model called Universal Data Model, UDM. And so when you create a repository there, you're looking really more at a flow of disparate assay data coming in, and then all the auto checks and auto mapping are set up and configured up front, and you have pretty much in real time, within just a couple of hours, the turnaround of wide variety of assay data that becomes immersive and analysis ready. And I think that the advantage to that is that if you're dealing with data from 2017, you can quickly realize that in 2023 or onwards, there are new domains that have come about that guide the standardization of certain collected data. And that may look very different in 2029. And so the idea that any data that's been collected can be standardized to any SDTM IG of your choosing, whether for analysis in the traditional and conventional and stats programming methodology and way, or whether for submission and regulatory submission, including submission of earlier performed analysis and assays. I think that's really where the value add is. And so for that sponsor specific model, we do have XBiome as a subscription on a SaaS basis. But for the more transactional CRO or lab oriented customer data set, sometimes that's transactional more at the assay specific level. So hopefully that answers your question. And if there are any more specific questions, once again, hello@pointcross.com. And that brings us to, we have a few more questions that I unfortunately think we've run out of time for, but we will respond to in writing. But I do want to thank everyone sincerely for having spent the half hour with us today. And again, we look forward to welcoming any questions or follow-up from this. So thank you all and have a wonderful day. Appreciate it, Arpita. Thank you.
Thanks, everyone.